Natalia, welcome to this seminar. I was really fascinated by photobiology back then. And that actually made me choose for biophysics as a specialization because I was really fascinated by applying light in order to understand, to study biological phenomena. So since then, biophysics and actually photobiology and application of light was very, very close to my heart. And I keep working on this topic even now. And then indeed, as you mentioned, I really wanted to do a PhD. I really wanted to pursue my career in science. And I moved to the Netherlands. All the way from Belarus, I moved to the Netherlands with 2 suitcases in order to study at the University of Twente. I did my second master there, and I did my PhD there as well. And then with a PhD here in the Netherlands, working for Philips research laboratories was really a dream job. And I was thinking, I only wish I could have a job with Philips. And I was lucky. I was very lucky. I got a job with the Philips research laboratories, and I have been working there really with a lot of passion and pleasure for about 15 years. And 11 years of that time I spent hand in hand in the lab with Dr. Babu. And you know, people always say that it is good to change your jobs. It is very good to go somewhere else and try a new job because you get new experience and you also better understand yourself. What is it that you truly like to do? What is it that you are really good at? So I decided to do a very big change. So from Philips, where I was scientist and I was working on biomedical applications of light, I moved all the way to ASML, to semiconductor industry. I was working on light sources as a project manager, but it was completely different industry, semiconductor, not healthcare. And I was no longer a scientist. I was a project manager, managing other people, managing schedules. And that gave me a lot of insights. So I really appreciate the choice that I made then and 2 years that I spent with a completely different company. Also, I understood that actually my true passion is innovation and science. So it helped me better understand myself, better understand my drivers and my capabilities and my passion. So I moved closer to science. I moved to Eindhoven University, but also I opened up my consultancy, BioLight consultancy. So I continued working on what is really close to my heart, biomedical optics and photobiology. I also became advisory board member in a startup that is working on photodermatology, dermatology using light. And actually this year I saw a really great vacancy in a whole center. And that was really about shaping the program on medical wearable devices. And I could not withstand resistance. So I applied for this job and I took this job. And here we are. So in the course of the years, I changed quite a few jobs, and it helped me understand different industries, different work environments. And it also helped me to understand much better myself. Who am I? What are my true values? What is my passion and where I'm good at? So dare to take changes, dare to take changes, dare to change the area of specialization, move around, and it's very useful. That's one of the lessons I learned. So if I really look backwards and if somebody will ask myself, hey, what is it that you're proud of, Natalia, when you were working for Philips? One of the things is that we've been working on a Philips product called Philips Lumea, and that is photoepilation, removal of hairs using light. And what our team did, really extraordinary, we really understood how this device is working. And we presented it at the international conferences. We published it in peer-reviewed papers, and really we underpinned the action of this product. Another one that I'm really proud of is we identified what is sensitive skin. A lot of people complain, oh, I have sensitive skin. I respond strangely to this cream or to shaving or to other things. But what is it exactly? Not really known. And we, together with medical doctors with dermatologists, together with 2 PhD students, we gave a better understanding what is sensitive skin. We published it in 10 publications. We wrote 2 book chapters about that. We gave many podium talks. And if you ask me, why, Natalia, are you proud of this? Well, this is another example where I really dared to make a change. I was a hardcore physicist and biomedical optics person. And to do this project, I really had to step out of the comfort zone. And to work together with dermatologists, I had to dive into skin biology, which was a very new topic to me. But it gave me opportunity to learn new field, to extend my expertise, and to also extend my fingerprint in this world. So therefore, I really appreciate this. So we had a wonderful European project together with a skin biologist from UK. We discovered very new light receptors in the skin and hair follicle. About this topic, I will talk in a few minutes in details, but from now on, you can take a message that actually our skin and hair, they can see light, just similar to how our vision is working. And this was absolutely extraordinary finding. Again, we published it in many articles. We presented at conferences, and we got 9 awards, international awards and nominations for this topic. And again, this is an example where we combine physics and biology, completely different fields, and that made a lot of impact. And also, yes, I participated in product launches of Philips, given scientific talks in Taiwan, in Japan, really assisting the product launches, translating scientific findings into practical implications, practical propositions for the products. And then in the course of the years, of course, I worked for several companies like Philips, ASML, like Eindhoven University, and now I'm with the whole center. And of course, through the years, you work with a lot of different partners in biology, like universities and medical centers, but also partners, for example, in material suppliers, or even a data steel, completely different beast. So when you change the fields, you also extend your network and you extend your knowledge, and also you extend your fingerprint or impact in this world. So dare to expand. Now, yes, Babu already mentioned, we published a lot, more than 120 peer-reviewed publications, a lot of patents, and myself and our students, we got 17 awards and international events. So that was great. And my passion was biomedical optics, photobiomodulation, and this is still my passion. I'm really happy I can talk to you about it. Now, when I look backwards, the very first project that I got at Philips was working on detecting blood glucose, detecting blood sugar non-invasively. And then we had a very new principle called variable multivariate optical element, complicated title. But this is my very first setup that I built myself from scratch when I came to Philips. I only had an empty optical table, nothing else. And I had to design this setup. I had to select all the optical elements here. And I had to build this Raman spectrometer using this variable multivariate optical element. Something I was extremely proud of, because until then, I didn't build complete optical setup by myself, and I've done it. So that was a great experience, hard job, but yeah, very good reward. And we published this data, and we also applied for patents based on this very first setup that I made. And then we moved on with biomedical optics. Here you see the setup that we constructed with the 2 optical fibers that were able to tilt. And by tilting these optical fibers, you can better control the depth to which light penetrates in the tissue. And yes, here you have the image when we were working together with Babu on polarization sensitive skin imaging. And you see here very complicated setup that we constructed, and we've been doing a lot of measurements on biological objects such as hair. And therefore you see here a lot of hairs on the optical table. And yeah, this is me and this is Babu. So my hair was different. So this is another thing that I was changing in time. So you can barely recognize me, but it's us working in the optics lab. And then the complexity of setup was growing and growing. Here you see an example of polarization sensitive optical coherence tomography. In those times, that was the first setup that we built in a lab. Such a polarization sensitive OCT was not available on the market, and our students, together with us, built it in the optical lab. And this setup, maybe Babu talked about it in the morning. This was a very complex, specially resolved polarization measurement in order to quantify polarization in a very special optical element. And again, back in those times, such a setup, such a device was not commercially available. We really needed it for our research. So we decided we build it ourselves together with the students in the lab. Now, enough of very small examples. What I would like to talk to you in the remaining 1 hour, 40 minutes, is actually photobiological action of light. Now, if you look at light tissue interactions include photochemical reactions, photobiological, photothermal, photoablation, and plasma-induced ablation. And these light tissue interactions, they're actually defined by power density that is falling on the tissue and the pulse duration. And very typical example that you know perhaps for plasma-induced ablation is cornea resurfacing, so the correction of the vision. For photoablation, very typical example is skin resurfacing, wrinkle reduction to get a smooth and tight-looking skin. Photothermal action of light, very good example is light-based hair removal or photoepilation. And I will talk about this in a second part of this lecture. And for now, we will focus on photochemical and photobiological action of light. And this process occurs under very low energies, very low energies. Now, if you look at us, if you look at human beings, actually, we as humans, we evolved under the influence of sunlight. We are continuously exposed to the sunlight. And our predecessors, they've been hunting, they've been working in the fields all day. So all day they had influence of sun on their skin. And actually, because of that, we as humans, we developed mechanisms in order to use this sun energy, but also to protect ourselves from very big amount of sun energy. And what are the examples? Well, obviously vision, we can see objects around us, so we respond to the sunlight, to the light, and we developed vision to navigate. Another very important example is production of vitamin D. Vitamin D is essential for the bone health, bone density. It's also essential to keep us in a good mood. And you see that people who move to Northern Hemisphere, for example, Denmark or Finland, where there is lack of sunlight, very often population there has depression and anxiety. And this is because there is not enough sunlight and people do not produce sufficient amount of vitamin D. So that's essential function. Obviously, melanin production of pigment in the skin. If I would be spending 4 weeks of holidays in Spain, I would come back nice and brown. So my skin would be responding to the sunlight, protecting me from excessive amount. And there are actually more mechanisms that humans develop in response to sunlight. So bear with me. Now, obviously, you know that the solar spectrum, it includes extreme UV light that is used at ASML in the semiconductor industry, but also UV, blue, green, red, and infrared radiation. And the blue light is a very interesting one. So actually, it has a few sub-bands, blue-violet, blue, and cyan light. And the blue light you will learn it today is very important in applications of light for skin treatment. Now, if we look at the sunlight, and you know it obviously, the sun spectrum changes during the day. In the morning, the sun has one color. In the bright noon, it has another spectrum. And later in the evening, you see very reddish spectrum of the sunlight, and it's not so intense. So sun spectrum changes during the day, not only in terms of the total power density, but also in terms of spectral distribution. And one extraordinary thing is actually here in the blue region that the sun spectrum changes most during the day. So we must have responded to these changes in blue light by developing specific mechanisms to translate these changes into physiological reactions. And yes, indeed, I give here a very long table that you can study after this lecture. You get the table, you can study it. So these are all examples of photochemical reactions that are happening in our body in response to the sunlight. All different classes. For example, cross-linking of DNA molecules, or formation of thymine dimers. It's a DNA damage. Or photoreduction of FAD. And one important photochemical reaction that is happening under influence of sunlight is actually vitamin D production. And the most important one that I would like to accentuate today is a light reception in the eye, human vision. This is happening by opsins, photoreceptors called opsins. But bear with me, this kind of reaction is not only happening in the eye, this light reception is also happening in our skin. And this is really something extraordinary. So long table, you study tonight, and I move forward. So actually our skin, if you look at the body, our skin is the largest body organ, and it is exposed to the sunlight. So obviously it is probably able to see the sunlight and respond to it and produce physiological responses. And we had multiple questions. So actually, how the light impacts the skin and hair in the skin, what would be the mechanisms of this interaction, and can we maybe translate this knowledge into useful therapeutic applications of light? And for that, we wrote European grants. So we've been working very hard. We wrote European grant, which is called Classic, and that was collaboration between Philips and University of Bradford Center for Skin Sciences. And I can tell you now, we wrote a lot, we published a lot of articles, we gave more than 13 talks, and together with our PhD students, we got 9 awards and nominations, including Best of Photobiomodulation Award in San Diego in the US. So what this project was about and how they started. Now, actually, if you look now at the market, there are a lot of light-based devices based on photobiomodulation or low-level light therapy. Devices that emit relatively low level of light. And these devices nowadays are on the market, approved by FDA, Food and Drug Administration, for treatment, for example, of chronic wound or treatment of hair loss. Now, however, if we look at the parameters of these devices, there is no consistency in the optical parameters. From one device to another, from this hair comb to this helmet emitting light, there could be between 10 to 40 times difference in optical parameter, and still they claim the same action. So something we thought is not okay here. Something is not accurate. It cannot be that 2 equal devices emit 40 times different parameters and have the same action. We decided to dive a little bit deeper into this topic, and we performed very big literature review on photobiomodulation and optical parameters for photobiomodulation. And what we learned actually that all the parameters that are reported in literature, they vary by at least 2 orders of magnitude, and still they claim the same efficiency when applied on the human body. So we also thought something is not okay here. It needs investigation, it needs clarification. And all this knowledge we published. We published an article in Experimental Dermatology, photobiomodulation devices for hair growth and wound healing, a therapy full of promise, but a literature full of confusion. So very sharp title, to the point. And we said that there are a lot of knowledge deficiencies that do not allow us to progress in this field, to develop better devices. So what we did different? We did it different. What you typically see in many fields, that everybody has certain specialization and works in isolation. Cell biologists, molecular biologists, they work together with biologists. Biomedical photonics people, they only work with biomedical photonics people. And photobiologists, they do a lot of experiments in a test tube, looking at protein conformation changes, but they don't talk to people from biomedical optics, and they don't talk to people who are busy with molecular biology. So we said, no, we have to change it, and we need to undertake a system engineering approach to photobiomodulation. And that basically means combine these 3 different expertise together. And all 3 expertises are needed to really make a breakthrough in the field. So put together in one project cell and molecular biologists, biomedical photonics people, and photobiology people. And really understand how a photon of light will interact with the skin cells, which molecular mechanisms are triggered, and how to translate this fundamental knowledge to something useful for the product. Now, if you look at what kind of molecules, what kind of photoreceptors or acceptors or absorbers can interact with light in the skin, there are many. So cytochrome C oxidase, protoporphyrin, NOS, NADPH, but also very interesting molecules, cryptochromes and opsins. Cryptochromes is a photoreceptor, and they govern circadian clock, our day and night rhythm. Many processes in the body, or nearly all processes in the body, they're different during the day and during the night, again, responding to the sunlight changes. Opsins, you know it from human vision. Opsins are present in the eye, and they enable vision. Now, opsins might be present also in the skin, and these are also very interesting molecules. So in our project, we focused on these 2, on cryptochromes and opsins, because they were very new, very new. And I tell you already now, you don't need to wait until the end of this lecture. I tell you already now that we discovered something very extraordinary. Actually, our skin acts beyond the skin barrier, beyond being the first line of defense, protecting us from mechanical damage or sun exposure. Actually, human skin can see light, it can feel. Human skin can also smell, because scientists demonstrated that if you take sandalwood oil and you put it on the skin, there are receptors of smell, and they respond to this sandalwood oil, and that helps actually to heal the wound. And also, human skin can produce pleasure hormones, endorphins. So human skin is fascinating, and it can see, feel, and smell. But how did we discover that it can see light? So as I told you, we focused on cryptochromes, which are regulating circadian clock, day and night processes in our bodies. These cryptochromes are in the cell nucleus. and opsins, which are transmembrane proteins. And there are many different types of opsins. There is opsin called rhodopsin, the same as in the eye. There is encephalopsin, the one that is present in the brain, actually. There is a melanopsin, also participating in circadian clock, day and night clock. There is also neuropsin, many different types. And what we did, we looked at the skin and at the hair follicles, and we tried to understand if our skin and hair follicles, they have these photoreceptors. An extraordinary thing, yes, our hair follicles that we have in the skin, they have these photoreceptors, they are present there. And another extraordinary thing is that our skin, the skin cells, they have cryptochromes and opsins, photoreceptors that can potentially respond to light, to the sunlight. But remember, as I told from the beginning, actually, we don't know what would be the beneficial parameters. Yes, there are photoreceptors, but what are the parameters of light that are needed in order to trigger these receptors, to trigger physiological reactions, to trigger useful reactions? So there was a mess in literature, nobody knows. And yeah, actually, there are many photochemical reactions. I showed you the whole table of photochemical reactions that can occur upon action of light. And these are all different classes of reactions. And obviously, any chemical reaction goes by its chemical law, reaction speed, reaction constant, equilibrium constant. And obviously, every reaction would need a different parameter of light to achieve a certain effect. But which parameter? How do we know it? What is even more complex is that you see that a lot of experiments on photobiology and photobiomodulation, a lot of experiments are done in a cell culture. So you take a single layer of cells, 20 micrometer thin layer of cells, you shine a light on them, and you investigate biological response. And yes, of course, in this way, you can find light parameter that is useful, that is beneficial, that has positive impact on the cells. But when you would like to make a device that actually acts on human skin has finite thickness. It is 5, 6, 7, 10 millimeters thick. And when your target, your cell is located at a different depth in the skin, how would you know how much light do you need to apply in order to trigger useful response? Because this is very thick tissue layer. Light will be absorbed, light will be scattered, light will be attenuated at a certain depth. So even if in a lab, using this very thin layer of cells, we can find a useful parameter, it's not obvious what should we use on the skin in order to achieve the same positive effect inside the skin. So how to solve this question? Obviously, what is actually needed is to know how light propagates in tissue. And there is a method called Monte Carlo modeling or Monte Carlo simulations. And this method can predict light absorption and light scattering in a turbid medium such as skin. And basically, what is then done, you need to know the distribution of light from the light source. You need to know the optical parameters of every layer in the skin. What is the absorption coefficient? What is the scattering coefficient? And then using computer model, you can calculate how the photons of light will be propagating in depth through the skin. But as I said here, you really need to know what are the optical parameters of the skin layers. And if you look at literature, there are so many publications reporting optical parameters. However, the reported data vary a lot. again, there is a massive unclarity, massive variability. So the Monte Carlo model we made, and this Monte Carlo model models the light beam, excuse me, the shape of the light beam. And we model the skin as three-layer structure: epidermis, dermis, and subcutaneous fat. And each of these layers has optical properties: absorption coefficient, scattering coefficient, anisotropy of scattering, and refractive index. And we need to know these properties accurately in order to model the process accurately. So what we did, we dived in the literature and we reviewed many scientific articles. And then we screened them and we selected 3 data sets for further analysis in order to understand which of these parameters are the most accurate, which of these parameters most accurately represent optical properties of human skin. And then when we use these optical parameters and when we run the Monte Carlo simulation, what you get as the output is photon density as a function of depth. And what you see here, if we use data set number 5, or if we use data set number 2, the photon density that we calculate is different by 4 orders of magnitude. 4 orders of magnitude. So there is a great variability in reported data sets, and it's not helping us to make accurate calculations. So we have to understand which of these data sets are most accurately representing human skin, so that we don't have these 4 orders of magnitude difference depending on who published an article. So we did a critical analysis. We published this, how we analyze it exactly. And we came up with the 4 most trustworthy data sets. And you see, if we use them, if we use the data from Steve Jacques, from Simpson, from Meglinsky, if we use data sets from Gregory Altshuler, and if we run a Monte Carlo model, then all these data sets give us nearly the same values of photon density, and which is great. So we identified most trustworthy data sets, and now we can predict light propagation in the skin. And that means when we do experiments on the cells, on a single layer of cells, and when we find a very useful parameter, we can translate it to the optical parameter that has to be used on a full thickness skin to achieve the same beneficial effect. But there were more complexities on the way, even more complexities on the way to solve. Actually, if you look at the experiments where we use light on human cells, light on biological tissue, obviously there is a variable such a wavelength. So one wavelength can have beneficial effect and another one not. And obviously irradiance and radiant exposure, or dose, they may have effect, but there are more factors that will impact the outcome of experiment. Biology is complicated, and photobiology connecting photonics and biology is even more complicated. So there are more factors. For example, cell density. You see here cell culture, and you see less cells in one case and more cells in another one. And cell density may impact the outcome of experiment. Cell lineage, cell type, different cell lineages, different cell types will respond differently to the photon of light, even at the same optical parameter. Environmental factors, for example, concentration of oxygen. Concentration of oxygen inside the skin is not the same as in the air. And it will vary depending on the depth of the skin, because oxygen comes through the blood vessels and then it diffuses and it goes to the skin cells. There will be even a gradient of oxygen inside the skin. And carbon dioxide, CO2 level will also be different, and even more parameters like cell culture medium and serum. So how to deal with that? And actually, if you look in literature, most of the researchers, what they do, they use one optical parameter, one wavelength, one dose, and then they fix these cell culture conditions. And in this way, they don't study the full parameter space because there could be a lot of dependencies there. But this parameter space is enormous. So how to deal with that? Design of experiment, there is a methodology called design of experiment that actually allows us to study these very large parameter sets and to identify factors that have the most impact on the outcome of the experiment. So we adopted this design of experiment methodology that is very well known in semiconductor industry, for example, or very well known in automotive industry to get the quality. We adopted it for experiments in photobiology. We were the first ones to do that. And yes, with all this in mind, so we developed really the very rigorous framework to study photobiomodulation. And we published all these articles in scientific papers. We published the article on variability of optical parameters. We published the article how different conditions of the cell culture may impact the experiment. And we published a chapter in the book called Molecular Dermatology. We published the whole chapter on our framework to study photobiomodulation. What is the right experimental approach to get the best out of your experiment, to cover the whole complexity of the field and to get a simple answer. What is also very important to mention that, yes, we identify photoreceptors in skin and hair follicles, and these photoreceptors, they have functional significance. That means they're not just there, but they really have functional significance. For example, the cryptochrome 1 is responding to blue light. Remember, I told you at the beginning of the lecture, have in mind blue light because it becomes more and more important for therapeutic applications. So photoreceptor cryptochrome responds to blue light in human skin cells, and this has positive impact on hair growth. So maybe somebody of you can come up with a therapeutic solution with a device that is using blue light and can stimulate the hair growth so that everybody has nice, long, beautiful hair at all ages. Also, what we saw is that blue light has impact on skin barrier function, and it may have useful applications for wound healing, for closure of wounds. And yes, also, I told you that we found opsin photoreceptor present in the hair follicle, and that opsin photoreceptor also has potential to have positive impact on hair growth. So again, take the opportunity, take this research line further on, and understand maybe you can come up with a device that has positive impact on hair growth using different parameters of light. And more. So the takeaway messages from this project actually that, yes, our skin is the largest body organ, and our skin and hair follicles that are present in the skin, or here, hair follicles, they have photoreceptors, molecules that receive light. And these photoreceptors are cryptochromes and different type of opsins. The cryptochromes, they are known to be circadian clock regulator. They regulate our night-day physiological reactions. And also we have shown that these cryptochromes can interact with the blue light, and that has implication for hair growth. And opsins. Opsins, they also interact with blue light, and they may have positive implication for hair growth. We also showed, and I don't tell it today, we also show that actually light has differential impact on human skin. What it means is that the impact of light depends on the wavelength. So blue light has one type of interaction, red and infrared another one. And also light has dose-dependent impact on the skin. Depending on the dose, either you can stimulate cells, increase metabolism of the cell, or you can suppress the cells, decrease metabolism of cells. For example, decrease collagen production. And positive and suppressive, so stimulatory and inhibitory effect of light may play a different role depending on application that you choose. For example, remodeling of scar, if you don't want to have a scar tissue, maybe you want to suppress the cells that are producing this extra collagen, and that will help to reshape the scar. And blue light, so remember the blue light. Blue light also has differential impact on the skin and hair cells. And the low doses of blue light, they have stimulatory effect, and it has positive impact on hair growth. Now, I shared with you a lot of scientific information that we did. So we identified receptors of light in the skin. We made a Monte Carlo model how to calculate light propagation in the skin in order to predict parameters. I showed you some of the experiments where we proved functional significance of these receptors, that they respond to light and they trigger physiological reactions, for example, hair growth or wound closure. Actually, there are many more applications of light in dermatology. And blue light from 400 to 500 nanometers, it takes a central role in applications for photodermatology. Blue light has been shown to have positive impact on psoriasis, chronic skin disease. It also has positive impact on reduction of eczema or atopic dermatitis, hair growth, wound healing, itch. Reduction of acne has been proven. So acne or pimples respond very nicely to blue light. So the one who has pimples can have very clear and nice skin by using blue light. Reduction of blood pressure. So blue light has positive impact on reducing blood pressure, can be very useful for hypertension, treatment of hypertension. Blue light actually releases endorphins in the blood, and endorphins, these are hormones that create this feeling good. So it can be very useful for example, treatment of depression or anxiety or bad mood. Blue light has potential for scar remodeling and even more. So there are a lot of applications. And so, yeah, what is then the future? And we think, together with medical doctors, together with the scientists, we think that the future is in whole body devices based on phototherapy, based on light-emitted diodes, and also wearable devices for therapy. And examples could be, this is a flexible wearable LED device that can be used on the face. This is the full body device combining different wavelengths of light, blue, red, infrared. This is one specific example for therapy of arthritis and pain that comes with arthritis. Flexible device for treatment of shoulder pain, and also flexible wearable devices for photodynamic therapy. So photodynamic therapy is the one that is used for treatment of cancer. It's a combination of active ingredients, for example, ALA and light, and together that helps to kill cancer cells. So we do believe that the future is bright for photobiomodulation, the future bright for phototherapy when treating dermatological conditions, but also systemic conditions such as blood pressure. And this is the end of part one. I show here a very beautiful landscape of North Brabant province where myself and Babu live. And if there are perhaps a few questions now, I can answer them. Up to you, or we take a little pause.